After studying this module, you shall be able to know the principle of ion exchange chromatography, learn the two different kinds of ion exchange chromatography, learn the considerations behind choosing an ion exchanger for an experiment, identify what kind of ion exchanger and pH conditions to use for separating a protein from a mixture, and lastly, how to analyze the results obtained from an ion exchange chromatography experiment. To introduce you to the ion exchange chromatography concept, ion exchange chromatography is a biochemical technique widely used to separate molecules on the basis of their net charge. This module describes the basic principle behind the working of this technique. It also introduces the reader to various kinds of materials used as resins that make up the matrix bed and the various ionic functional groups that are bound to the resin. Pages discuss the various features that characterize different ion exchangers and the considerations to be kept in mind while selecting an ion exchanger for an experiment. Lastly, it briefly outlines a couple of useful applications of ion exchangers. We'll now be discussing the principle of ion exchange chromatography. Ion exchange chromatography is a technique used to separate proteins on the basis of the net charge carried by them. The magnitude of this net charge carried by the protein depends on the pH. Ion exchange columns consist of a solid polymer, which is a resin, onto which charged groups are bound. The solid matrix or resin can be of different varieties. These include matrices made of polystyrene, cellulose, agarose. The matrix can also be made of a copolymer of styrene and vinyl benzene, in which divinyl benzene cross-links the polystyrene and also carries the charged groups. A resin to which anionic groups are bound are called as a cation exchanger, and a resin to which cationic groups are bound is called as an anionic exchanger. As you can see in this figure, the left hand side shows an anion exchanger and the right hand side shows the cationic exchanger. Some groups are more strongly ionic than others and are referred to as strongly cationic or anionic, while some groups are weakly ionic and are referred to as weakly cationic or anionic. Sulfonic group SO3- is a commonly used strongly acidic cation exchanger. If it binds an H+ ion, it is said to be in its acid form. It can exchange one sodium ion for a H+ ion and one calcium ion for two H+ ions. Phenolic and carboxylic groups are example of weakly cation exchangers. A quaternary amino group is a strongly basic anion exchanger. Aromatic and aliphatic amino groups are weakly anion exchangers. The table shown on the lists the various types of resins, the functional groups that serve as the ion exchangers, and their commonly used trade names. Ion exchange chromatography works on the principle that charged molecules will adsorb reversibly onto the column and will be eluted by changing the ionic environment. The molecules in the sample mixture are first allowed to tightly bind to the column. Appropriately changing the buffer pH or altering the ionic strength of the buffer allows elution of the bound substances. The order of elution of substances from the column depends on their affinity for the charged groups on the column. For an anion exchanger, the cationic species in the sample mix will not bind and hence will be eluted first, followed by the anionic species. The diagram on the screen shows you how a negatively charged protein and a positively charged protein is separated on an ion exchange column. Cations will not bind to an anion exchanger and elute first, followed by the anions. Converse would hold true for the cation exchanger. Also, weakly bound substances will be eluted first, followed by substances bound more strongly. Affinity of a bound substance to the resin will be a function of both the pH and ionic strength of the buffer system used. Now we'll discuss the considerations when working with ion exchangers. Firstly, the features of an ion exchange resin. There are some important properties of an ion exchange resin that need to be taken into consideration when deciding to use an ion exchanger for separating molecules. These are total capacity of the resin, available capacity of the resin, its porosity and its mesh size. Total capacity of an ion exchanger is the total milli equivalence of exchangeable groups per milligram of dry weight. It's important to not exceed this capacity, otherwise the ions will simply pass through the column without binding. However, experimentally, the ionic groups actually available for binding depend on the pH and ionic strength of the buffer being used. There is always competition between the small buffer ions and the charged macromolecules for the ionic groups on the exchanger. Small ions are able to diffuse rapidly through the charged groups of the exchanger and hence compete with the large macromolecule. This competition becomes more enhanced as we increase the ionic strength of the buffer. The capacity of the resin that is actually available under the conditions of the experiment is called as the available capacity. Now we'll talk about the porosity of the matrix. The charged groups on the matrix are both inside and outside the matrix. The beads of the matrix also act as a molecular sieve. In case of polystyrene based resins, the size of the pores are determined by the extent of crosslinks, and that is determined by the amount of divinyl benzene. For example, for Dowix and Ag matrices, the percentage of divinyl benzene is indicated in its label. Dowix 50x8 for example is 8% divinyl benzene. Greater crosslinks increases the capacity of the resin. Small molecules are better separated on matrices with small pore sizes. Macromolecules require larger pore sizes. Cellulose ion exchange resins are fibrous and are best for purifying large molecules like polynucleotides and proteins because their functional groups are exposed on the surface and hence easily accessible for binding. Beaded forms of cellulose resins are also available, such as diethyl aminoethyl, Cefakel and diethyl aminoethyl biogel P resins. Such resins offer molecular sieving effects and allow better flow rates, which improves separation. Besides porosity, the size of the beads, particle sizes also affects the separation of substances on an ion exchanger. This is called as mesh size. Greater mesh size implies a smaller surface to volume ratio and hence decreased capacity and more time taken for an exchange to occur per unit volume of the resin. This leads to slower flow rate. More time taken by a substance on the resin allows for greater diffusional spreading that compromises the resolution of the separation. Using a finer mesh, on the other hand, will cause less time for an exchange on the resin and hence better resolution. Now we'll talk about the choice of an ion exchanger. The very first choice to be made when performing ion exchange chromatography is to decide whether to use an anionic or cationic exchanger. In case of a mixture of proteins, the net charge on the protein will determine whether they will bind to an exchanger or not. The net charge on a protein depends on the pH. Hence, knowing the stability of the proteins in the sample mix at the pH being used in an experiment is very important. Proteins are stable over specific pH ranges. If a protein is stable at a pH which is above its isoelectric pH, that is its pi, then it will be negatively charged. A protein that is stable at its pi will be positively charged. The former will bind to an anion exchanger, while the latter will bind to a cation exchanger. The figure displays this concept. As you can see in the figure displayed on the screen, we have the x-axis showing the net charge on the protein and the y-axis showing the pH. The middle dotted line shows the isoelectric point. All the proteins that are stable isoelectric point, that is up till pH 5, will be strongly bound to the cation exchangers, while the proteins that are stable above the isoelectric point, that is from pH 5 to pH 10, will be bound strongly to an anion exchanger and can be used accordingly. Either of the two kinds of exchangers can be used for proteins that are stable over a wide pH range. A priori knowledge of the stability of proteins in our sample mix helps to make a wise choice of the exchanger to be used. The next choice to be made is whether to use a strong or a weak exchanger. Since strong exchangers operate over a wide pH range and weak analytes require a very low or a very high pH for ionization, separation of weak analytes can be achieved on a strong exchanger. Similarly, weak exchangers will be suitable for binding strong electrolytes. Weak exchangers are advantageous not only because charged impurities will be less likely to bind to them, but they are also less likely to cause protein denaturation. Also, weak exchangers allow better resolution of substances, the charge differences between them which are small. Strong charge carrying biomolecules will bind too strongly to a strong exchanger, making elution a difficult task, and therefore a weak exchanger will be once again the exchanger of choice in this situation. Thus, a weak exchanger is more advantageous than a strong exchanger. Strong ion exchangers are needed when working with macromolecules that are unstable at low ionic strength. In such cases, high charge density on the ion exchanger is desirable to enable the binding of the macromolecule at high ionic strength. The Cefidex and BioGel exchangers are suitable resins of choice for these macromolecules, as the density of ionizable groups on these resins can be made several times higher than possible with cellulose ion exchangers. Now we will talk about the selection of buffer, pH and ionic conditions. Anion exchangers will utilize the cationic buffers such as tris, pyridate, alkylamines, and cation exchangers will utilize anionic buffers such as acetate, barbutyrate and phosphate. The rule is used while selecting the buffer to be used in an ion exchange chromatography experiment. Cationic buffers with anionic exchangers and anionic buffers with cationic exchangers. As a rule, the pH of the mobile phase buffer must be chosen in such a way that it allows the charged groups on the matrix to remain in their ionized state. In case of a cation exchanger, the charged groups on the resin should be maintained in their anionic state. This will happen only at pH values above the pKa or the dissociation constant of the charged group. Furthermore, binding of the sample molecule to this cation exchanger will take place only if it is positively charged. For proteins, this will be possible only if the pH is below the isoelectric pH, that is below their pi. Thus, binding on a cation exchanger will be achieved only if the pH of the mobile phase is above the pKa of the charged groups on the resin and below the pi or the isoelectric point of the proteins in the sample. For example, in cation exchange chromatography, using a functional group on the solid support with a pKa of 2.2, a sample molecule with a pi of 9.2 may be run in a mobile phase buffer of pH 5.5. Similar reasoning applies for an anion exchange column. In anion exchange chromatography, a molecule with a pi of 5.8 may be run in a mobile phase buffer at pH 8 when the pKa of the solid support is 10.3. Generally, better resolution is achieved if the ionic conditions used to load the sample on an ion exchange column are similar to those used for eluting the column. Buffers with high buffering capacity are desirable so that the ionic strength need not be too high. Now we will talk about elution from an ion exchange column. Binding of substances to an ion exchange column depends on the electrical properties of the substance in question and those of the charged species in the solvent. Elution can be achieved by changing the pH in order to alter the charge on the bound substances, which in turn will affect their affinity towards the resin, or by competing out the bound substances using salts. On varying the pH, different bound substances will be affected differently, depending upon their respective electrical properties. Weakly bound substances will be eluted first, followed by elution of strongly bound substances. Two methods are employed when eluting an ion exchange column: a continuous ionic strength gradient elution or a stepwise elution. Selection of either method is specific to the situation in question and is usually arrived at by trial and error. The figure on your screen will show difference in resolution observed when using a steep versus a shallow salt elution. Notice how the resolution is considerably improved when a shallow NaCl gradient is used for eluting the column versus a steep NaCl gradient. As you can see on the graph shown on the screen, two kinds of elution profiles are shown. The left hand side shows the elution when a steep NaCl concentration is used, while the right hand side shows the elution when a shallow NaCl gradient is used. It can easily be seen that the resolution is much better when the steep NaCl gradient is used. At times, good separation is achieved if the binding and eluting times are the same. This is called starting condition procedure. The figure on the screen shows an example of an elution profile from an ion exchange column in which RNA nucleosides are eluted. Downstream usage of the separated substances should also be considered while choosing elution conditions. For example, in case of the substance being radioactive, care should be taken that the elution buffer should be free of any potential quenchers. Similarly, in case the purified substance needs to be spectrophotometrically analyzed, then the elution buffer should not absorb in the required wavelength range. If the substance is an enzyme, its stability should not be compromised during elution. Now we'll discuss the applications of ion exchangers. Ion exchangers, besides being used for separation of macromolecules, also have other interesting practical applications. For example, desalting. In order to separate ionic species in a sample, a solution of macromolecules, ion retardation resins can be employed. Such resins contain both negatively and positively charged groups and can therefore remove both cations and anions. Uncharged molecules will simply pass through the column unhindered. This way, the eluent will be free of charged species. Ag11, A8 and Biorad are examples of ion retardation resins. Biorad resin is also called as Chelix. It is a cross-linked styrene divinylbenzene resin which is linked with imino diacetate ions. This resin is useful in removing divalent cations from a solution containing a mixture of univalent cations. For example, if calcium ions are required to be removed and replaced by magnesium ions, the Chelix exchanger offers a simplified and a faster method compared to conventional method of achieving the same. Upon passing such a solution through a Chelix column, the calcium will be retained on the column and the rest of the solution will pass through. Magnesium can then be subsequently added to the eluent. Ion exchangers are commonly used also to prepare deionized water. Ionic species are removed from water using what is known as a mixed bed resin. They are strong acid cation and strongly basic anion exchangers that are mixed together and then used. In order to make deionized water, the cation exchanger is regenerated by using hydrochloric acid. The H+ ions from the acid bind to the negatively charged groups on the cation exchange resin. the anion exchanger is regenerated using sodium hydroxide. The hydroxyl ions from the alkali bind to the positively charged groups on the anion exchanger. The hydrogen ion does not have very strong attraction to the beads of the cation exchanger, and the hydroxyl ion does not have very strong attraction to the beads of the anion exchanger. This is why on passing ionized water through a mixed bed resin, cations can easily be exchanged for hydrogen ions on the cation exchanger, and anions can be easily exchanged for hydroxyl ions on the anion exchanger. The released hydrogen and hydroxyl ions combine to form water. Now we will summarize what we have learned so far. Ion exchange chromatography is used for separation of charged molecules from a complex mixture. Protein molecules are commonly separated on an ion exchange column. An ion exchange column consists of a solid resin to which charged groups are bound. The resin can be made of agarose, dextrin, cellulose or copolymers of styrene divinyl benzene. Cationic groups bound to the resin matrix constitute an anion exchanger and is used for separating anionic substances. The resin can be strongly or weakly acidic. Anionic groups bound to the resin matrix constitute a cation exchanger and is used for separating cationic substances. The resin can be strongly or weakly basic. The actual number of charged groups available on a particular resin depends upon the experimental conditions such as pH and ionic strength of the buffer used. This is reflected in the binding capacity of the column. Charge, binding capacity, porosity and mesh size of the resin are important features taken into consideration while choosing a resin. Ion exchange columns separate molecules on the basis of their net charge. For a given ion exchange column, binding affinity for a particular analyte depends on its net charge under the conditions of pH and ionic strength of the buffer system being used. Choice of an ion exchanger for protein molecules depends upon their stability at the pH being used. Strong electrolytes in the analyte are better separated on a weak ion exchanger. Substances that have small differences in their charge are better resolved on a weak ionic exchanger. Similar substances are best resolved using a single pH and ionic strength condition. Elution conditions need to be carefully decided depending upon the downstream uses of the purified analyte. Besides being useful for separation of small organic molecules, ions and macromolecules, ion exchange chromatography is also used for desalting, preparation of deionized water, that is mixed bed resins, and as a matrix for partition chromatography.